Okay, good morning, everyone on Zoom. I hope you're doing well and staying out of the rain. So I'm going to go ahead and get started. And today we're going to be doing the population stratification. So we're going to be looking at making PCAs, pruning for linkage disequilibrium and minor allele frequency, and making an admixture plot. So these are my favorites, so I'm kind of excited today. Hopefully you'll find it fun too. But before we get into that, let's review the exercise from yesterday. So for those of you on Zoom and in person, did you guys try and remove the outlying heterozygosity individuals? How'd that go? No? Okay, no worries.
I didn't realize you uploaded it on the Google Drive, and so I didn't.
Oh, the PowerPoint?
Yeah.
Oh, okay, yeah. For those of you on Zoom too, so after each class, I'm gonna upload the PowerPoint on the Google Drive. So you can always go back and look at the class.
I didn't catch it, but I found some code that was basically looking, it was trying to like get rid of 3 standard deviations outside, and so it was using awk to generate a new
Okay.
column with standard deviations, and then
Okay, so you did just like a Linux command, basically.
yeah,
Oh yeah.
I found a way.
No, that's great.
And then
Yeah.
that generated a new file with the IDs to be
Yes.
dropped, and then you could do it looking at to drop.
Yes. Yeah, that absolutely works. Yeah. How many individuals did it drop?
It was I don't know if I did it right. I think I had 380 at the end. It's like what was left.
380. Okay, so it may have dropped more in your case because you put in a number threshold. So it was like pruning based on that, and which I'll talk about in a second. I was more looking visually which individuals fall outside of it. So there's really no right or wrong answer. You're not wrong. Like you did it absolutely correctly. There's multiple ways to do it. It just kind of depends on how stringent you want to be with taking people out. Yeah. So, yeah. How about on Zoom? Do you want to does anyone want to share how they did it? Or if you weren't able to do it, maybe do a thumbs down? Oh, chat. Okay, Asha said she sorted Excel file by column F from largest to smallest, deleted 4 people with values greater than 3, and then you saved it as a text file, and then you filtered keep. And then, okay, so you kept everyone but those 4 people. Awesome. So you ended up with 384 people. Great. Yeah, that's the way I do it. I also think there's a command in Plink that's a double flag remove. So you could make a text file with the people you want to remove and then remove them as well. So the reason I if anyone else on Zoom, thumbs up, thumbs down, tried it or want to share, you can also type it in the chat if that's easier. I picture everyone at home just getting their morning coffee and like slowly waking up. Yeah, so the keep allele order is an important flag because, as it sounds, it keeps the allele order. So sometimes it can rearrange them if you're outputting in a different file format. So it doesn't hurt to keep that in there. And then the make bed makes it into a binary Plink file. So it outputs it in that like bam, bim, fam. So if you just want it to be a ped file and a map file, you would get rid of the make bed flag and just have the out flag. Okay, so we had a couple different ways of removing the individuals, and I had you guys try it on your own because there's a lot of different ways in Plink to do it, and there's a lot of ways out of Plink to do it as well. So sometimes it just boils down to personal preference and how big your data file is, right? If you had, instead of 300 individuals, if I had 10,000 individuals, I would do exactly what you did. I would do a Linux awk command to get the distribution and then filter it by a threshold, because those files are so large you can't open them in Excel and get a visual. So yeah, so the way I did it, which we briefly talked about in class yesterday, is I opened the .het file in Excel, and then I quickly plotted the frequency to kind of look at where the individuals were falling. And I saw that for the most part, they were within these two windows, which is 0.3 and negative 0.2. So I decided to trim these 4 individuals. And to identify those individuals, I sorted the F column exactly how Asha did, and I created a text file with the rest of the individuals. So everything but those 4. And then I basically repeated the exact same plink command we did yesterday when we were extracting those populations from the larger VCF file. So, and if you weren't able to do this step on the Google Drive, I've uploaded this output binary file. So I'm gonna put it in the chat, and then if you guys wanna look in your email, I'm gonna do like some quick background today. So go ahead and download that so we can do the rest of the steps together if you weren't able to do it. Yeah. I think. Okay, so for everyone on Zoom, this is where in day 2, you're gonna download these chromosome 7 underscore clean files, and that has all of our missing data removed. It has related individuals removed. It has outlying heterozygosity removed, and Hardy-Weinberg, those outside of Hardy-Weinberg are also removed. So this is what we're gonna work with today for population stratification. Okay, so if you did do it on your own, this is what I ended up with. You can work with the file you made too with less individuals. It won't affect anything we do today. But I ended up with a little over 4 million variants and 384 people. And going in, you can see there were 388 people. So it pruned 4. Or I should say cut 4, I shouldn't say pruned. Okay, and then how about the other 2 take-home exercises? Did you guys take a stab at doing those?
Just UK Biobank now?
Huh?
UK Biobank
Just thinking about it?
Or
Yes.
Or the UK Biobank.
Oh, the UK Biobank? Yeah, definitely. Oh yeah, that counts. Yeah, and then why would that be useful to you?
Hundreds of thousands of exomes.
Yes,
And
yes.
clinically connected.
And exome data is really easy to work with because the VCF files are smaller, so they can be higher coverage and smaller. So you can process a lot more people, which is nice. Yeah, okay, so for those of you taking the class for a grade, the take-home exercises are together gonna be your assignment. So they'll all be on the Google Drive. How about on Zoom? Did you guys have a chance to poke around the internet? Did you find any other public repositories? TCGA, what's that?
It's a tumor cell genome atlas. Yeah, tumor cancer. I mean, the cancer genome atlas. Yeah.
Very cool. I always like hearing about what people are using because you never know. Someone might have a resource that someone else in the class might find useful. That's really cool. Great. Okay. And then what I was showing everyone here in person, 23andMe. I don't know. Correct me if I'm wrong. Does 23andMe let you download other people's data anonymously? No.
They do research and like ask for your, they can use your data.
Yeah.
But I don't think they like.
I think you can request it, but it takes a while, right?
You can request yours, yes.
Yeah, well, you can get yours like almost instantly, I think, right? They just email it to you.
Okay, yeah, I don't
Yeah.
know how long it takes, but yeah,
Yeah.
it's definitely not publicly available.
I actually, sorry, this is a bit of a tangent, but I actually was at the American Society for Human Genetics last week, and I was talking to someone who works there, and apparently they impute your data. So they're basically inferring markers that they're not even sequencing. So they have all that information. They just can't release it to you because of FDA regulations. So when you do a 23andMe, they give you a lot of behavioral and like phenotypic traits that don't make sense for the markers they're testing. And I always thought that was kind of strange. And it turns out it's because they're basically like inferring your genetic data at other markers. Yeah. Yeah. But I feel like at one point 23andMe or something similar, hi, good morning, made data available. But this was like 10 years ago, so it might have been a different company.
And there was a one talk here where they were able to download other people's data by uploading a sequence that was like heterozygous for everyone, and then you match with relatives and kind of infer
The
what their
relationship.
genetics were. Anyway,
Oh, that's
we're sort of doing that.
interesting. Okay, so you basically get other people's genetic data, so
Yeah.
there's no privacy if you're related.
Yeah.
Right? Oh, that's interesting. For those of you on Zoom, I was showing everyone in person that the link to a website for Mac is still down. But in the future, if you are interested and want to poke around for that third take-home exercise, all the functions are here on the left, right? We did a lot of the basic statistics yesterday. So you can click on a lot of these and it'll give you all the different commands for each statistic, and sometimes some papers where they use them, which is really useful. Okay, so I'm gonna do some kind of basic background knowledge for what we're gonna do today, and then we'll take a break, and then we'll do hands-on, and then we will enjoy the rest of this rainy day. So while I am giving you basic background knowledge, did I put it in the chat? I highly encourage going to the Google Drive if you haven't already and downloading these files. This has the heterozygosity removed and we'll be working with it today. And it's in day 2. Okay, so today we're looking at population structure. And when we think about population structure, it's interesting, right? Because we're looking at very few or a very small percentage of differences between populations, right? So 90% of total genetic variation is found in a collection of individuals from a single continent, right? And 10% more variation would be found if the collection consisted of all human populations. So basically what that means is most of the variation we see is within a population and not between populations. And this idea came from Richard Lewontin, who kind of was a big founder of genetics and human diversity, and he used, or I should say, the theories of evolutionary genetics. And he used a lot of different serum proteins and red blood cell enzymes from different populations around the world to show this concept, right? So before him, everyone kind of just assumed different populations had more differences, which seems more intuitive, but is not the case. And when we think back to yesterday, one of the biggest caveats of Hardy-Weinberg equilibrium is that outside forces don't affect it, right? That there is random mating, that there is no genetic drift, that there's no population bottlenecks, right? You kind of assume everything is always neutral in the population, but we know in reality that is definitely not the case. The only kind of known example I could find, if you know more, please chime in, is of these like polyp spores in the ocean, and they release their, what does it say? They release their eggs into the water and then they float around and they get fertilized by chance, right? That is pretty much one of the only cases of random mating on the planet, I would say. I don't know. Does anyone know any more?
I think there's some fish that do that too.
Something
Spray out eggs and
similar.
spray out sperm.
Yeah, but then you have to assume there's no like sexual competition between the males, right? To get to the eggs first. Yeah. So it's kind of a big caveat, right? When you think about population structure, because you assume all these forces don't happen, but we know they happen. Yeah, so this will cause allele frequencies to be out of Hardy-Weinberg equilibrium. And we also know that populations become isolated for various reasons, right? Either geographically or maybe culturally, they're mating with each other for specific purposes and not a neighboring population. So all of these forces together basically result in what we call population stratification, right? It's what creates the differences within the populations. And for this workshop, we're going to use different genetic distance measurements to look at this population structure. So one of the first main genetic measurements is called cluster analysis. And there's a lot of different programs that do this. There's Structure is a big one. The one we're using is Admixture, but like all caps because they're fancy, right? And how these programs work is that you group individuals into different clusters. So these different clusters are represented by the variable K, which is just the number. And then this analysis uses, in admixture's case, a semi-machine learning algorithm to group the individuals logically according to the ancestral proportions you give it. So once you run that, the final coefficients that pop out reflect the probability of the individual belonging to those different clusters. And you could interpret these as fractions of the genome having membership, right? So if you do something like 23andMe, they'll probably run a similar program to this. I don't know exactly what they do. And then you get those percentages, right? I'm 30% North African, I'm 10% European, and this is kind of the same thought process. So this figure I've been showing is from the program Structure, and these individuals are from Pakistan, and you can see they subdivided, or they told the program to do 5 clusters, right? And then each column here, sorry, it's very small for you guys, is one individual. So those colored blocks going up and down are the proportions of ancestry that that individual has from those populations. Does that make sense? It'll become clearer. So when we apply this to other regions, right, you can imagine that, let me do this. Oh, I have a chat question. Individuals on x-axis. Yes, exactly. I'm gonna make it bigger because we're not coding right now. So you can imagine, right, if I'm looking at individuals from North Africa and I tell the program, okay, I want you to sort these individuals into 2 ancestries. It'll result in that chart on the top. And then you can tell it 4 ancestries, 6 ancestries, 8 ancestries. And the y-axis is the total percentage, and the x-axis, each bar is an individual. So you can see, depending on how many ancestries I tell the program to compute, the individuals have vastly different proportions, right? I would say especially when you look at the more admixed individuals, like say individuals from Egypt, 8 ancestries versus 2 is very different. And then another main analysis to look at population structure is what's called a principal component analysis, so a PCA. And this basically takes a lot of different variables and it reduces the dimensions so that it's more visually understandable, right, when you go to analyze the information. And it does this with a minimal loss of information. So have you guys seen this figure before? Yeah, this is one of my favorite figures. So this was a really nice paper that came out, I think it was Bustamante and all, 2014, I could be wrong, maybe it was earlier. But this shows they used a PCA to look at allele frequency data of populations in Europe. And you can see that when they overlaid the PCA to these different countries, it very nicely matched up regionally. And the thing also to keep in mind about when you plot a PCA is that the PCA is very much dependent on the populations you put in it, right? So in this example, we have 3 very different populations, and I mean different in that they've been geographically isolated for some time, so they've accumulated different variations from each other, right? So if you were to plot them in a PCA, they would turn out something like this, where the population clusters are very discrete and they're very far apart. But if you introduce a population that's maybe had some admixture with other populations, it'll result in a PCA like this, right? You'll have considerable overlap between the populations. So it's just kind of another thing to keep in mind of when people plot PCAs, right? What populations are they introducing to the PCA? And sometimes that can tell you more than what the actual figure is trying to tell you. So in this example, right, the individuals that show a considerable degree of overlap would be South Indians with, I think this is East Asians and Europeans. And that's because they have undergone admixture with these populations. And the technical definition of admixture is the formation of a hybrid population by mixing ancestral populations that have been in relative isolation. with the caveat that, of course, no original population is ever fully not admixed, right? We're all admixed to some degree. It's just kind of a relative speaking term when you think about populations on an evolutionary scale. So some examples of admixed populations are modern Europeans. Another great example is Puerto Ricans, right? They have African ancestry, they have European ancestry, they also have Indigenous American ancestry, and then African Americans, right? Mix of African ancestry, European, and even possibly Indigenous American. So this is kind of what we mean by admixture, where you can visualize if I were to plot these populations on a PCA, they would show overlap with these other populations that they have shared ancestry with. And we can also, if you're interested in this, you can also assess admixture using a PCA or admixture plot, right? So basically, let's say I have individuals from the Caribbean islands, right? And I want to figure out what proportion of African ancestry my individuals have. You can compute a PCA using reference populations from Africa, from Europe, from Indigenous Americans, and then see where your individuals fall on that PCA, right? Are they clustering more closely with the African Americans or another population? So that kind of gives you some inference as to their admixture proportions. And when you also think about admixture, this will disrupt linkage disequilibrium, and linkage disequilibrium, or LD, which I'm going to say because linkage disequilibrium is kind of a mouthful. LD is basically the non-random association of alleles. So when you think about admixed populations, they are going to have disruption of this, right? Because these two relatively different populations are going to have different allele frequencies. And then when they result in, for lack of a better term, a hybrid population, right? You're going to have these alleles in a new recombinant way that has now disrupted these previous LD proportions. So when you look at LD proportions in a population you know is admixed, it can help you to some degree infer the ancestries as well. It's just like a different method of looking at that. It also can help you infer as to the timing of admixture, right? So if you think about the previous example of like Puerto Ricans or the Caribbean islands, you know there's a colonial event that resulted in a horrible population bottleneck in, say, like the 1600s, right? So you know that from that point forward, the LD that population experienced is going to decrease. So if you know that, then you can time the frequency of the alleles in that population. Okay, so like I just said, it can help you date the origin of admixture events. So if you have known populations that have experienced admixture and you kind of know the allele frequencies, maybe you could use that as a foundation for figuring out a different population where you don't know when the admixture event occurred. So for instance, they are doing this to some degree with archaic introgression, right? So trying to figure out when ancient humans and like Neanderthals met up and had an admixture event, right? And then had hybrid humans. So this can help them narrow down that timing. It can also help when you look at disease. So if you're thinking about a disease causing allele or variation, and over time it's gonna decrease, right? You can plot that back in the phylogeny of the related individuals to kind of figure out what was the heritage of that allele, what population is it more associated with, and take it from there. Okay, so in summary, population structure affects allele frequency differences. So big forces, population bottlenecks, genetic drift, admixture events, and you have to correct for population stratification before doing a PCA or an admixture plot, because if you don't, it will lead to basically false signals, right? And you'll think you found something really exciting and it'll just turn out to be a marker you should have pruned, which is a bummer. And LD can be influenced by a lot of different factors, and you must always prune before doing admixture, PCA, or a GWA, right? So we'll talk about this a little more after the break, our first break. But there's a lot of different thresholds for how you can prune LD, but you should always do it. It's kind of a necessary step. So, okay, so we'll take a little break, and then how about like at 9:47, we'll do the next part.
Question that would benefit, the newcomers like myself. Currently we're in the shopping for textbooks.
Oh,
what
right.
would you recommend for one, concept, and then two, quantitative benefit for statistical? I think we're talking about that yesterday.
Yeah, and let me look at the other one.
This is so dense.
Nice.
It's really dense. It doesn't matter. Yeah, it's very, it's dense. Because when you do research, it's happening. The single content is bad, so
So I'm gonna be honest, I don't really learn that well from textbooks, so I don't
I
read
do,
them.
unfortunately.
Um,
I just think I like it for reference, like
Okay.
Yeah.
Yeah, well, I'm just caveating that. I've heard these are good ones, but I personally have not read them. So this one is, I've heard is good. The guy, that's the guy I was talking about. He's a population geneticist at USC. So he wrote this with genetics in mind.
Well, for statistics, besides statistics in general, the introduction to statistical learning is like the one.
Yeah. And then I think the other one you already knew, but it's a red cover and it has like bugs on it.
Yeah, but the thing is, it's so old, it's like 10 years old.
No, they have a more recent version, I think, right?
The one from yesterday.
This one?
Wait, what are
The
the books
second
called?
edition?
Oh, is there, what year was this one? I think it was,
It says 2004, but this is not, this is like the theory of.
oh, that's a theory, okay, not the quantitative aspect of it.
What was it called? This one.
Okay,
Are you talking like a book that
no, so
explains
this
like
is
methods?
this is theory, I'll get that one then.
So
I want
if
to get
you want
methods and
methods, that's
quantitative.
never gonna be the book.
Exactly one.
I would look
Check
for
this
review
one out.
articles.
This is really good. They update it all the time, but it's like heavy. Industrial stuff. Like this guy is actually people. It's like some
Who wrote the text?
It's like a good base. And then I think these two.
Wait, I need to know. These are good. I use these more when I work with insects, like these concepts. Josh Akey is like really good. But honestly, if you're looking for method stuff, I will look at articles. Because that's gonna be, Rasmus Nielsen is really good. The problem is with books is, especially book chapters, is that people, so you're not gonna get any stuff. So I would try and find a review art, like specifically a review article from like the last 2-3 years, and then see what methods they talk about for the teaching. So for instance, I'm thinking, I'll try and pick it up. There's a review article that basically talks about like all the selections you have to do for real frequencies versus haplotypes. And then they discuss like the pros and cons of each method. So depending on like what method you want to do, I think the review article focuses on that because then it's going to be the most yeah. They're never going to publish. So the idea of book is better for like background stuff. Theory. Yeah, like helping you think about how to use the method.
Why we're doing the brand.
Yeah. And then the other thing to keep in mind is for the most up-to-date programs, especially in genetics, they're usually really up-to-date, which is like no one has really used them, which is no one has really troubleshooted them, which means you're really on your own figuring out how to use them. So a lot of people still use methods that came out like 10 years ago because there's so much help online for everything. So yeah, so for instance, we're not doing that in this workshop. We're gonna do admixture, which looks at a population level, the ancestral clusters of individuals, but you can do local ancestry inference too. So if there's like a gene or a region that you're really interested in, you can look at the ancestral proportions of that gene for individuals, right? Like say a cancer gene, you want to find out if there are 100 people have different ancestries associated with like inheriting a recessive allele or something. They are pumping out a lot of really recent methods to do that, but they're so new. There's like no troubleshooting on it right now. So people are still using the methods that came out with it. It's just kind of, I mean, you know how PIs to get, right? They run something, they know how to do it. That's what they want to say. So, yeah.
For the linkage, just equilibrium, is
Yeah.
that like is it like transposons? Is that how like the alleles are associated with one another, sorry.
No, it's just kind of the way the alleles are coded.
It's because of recombination, right?
Yeah.
Remember like back to Mendel and centine organs and which is a probability of a recombination event happening between two genes. And so if genes are far away, there's probably get like high likelihood of recombination event. And if they're very close, they're unlikely to be separated by a recombination event. So it's just that
And
just because of that.
in addition to that, you have to think about like if there's some sort of selection effect going on, right? So if genes are non-randomly associated because one has been like they did together, like they're associated with the phenotypic outcome.
And that's where the disequilibrium come in, or that's
So the disequilibrium is basically just saying that anytime that theoretical balance gets disrupted, so anytime there is, instead of like random recombinant inheritance of alleles, it becomes non-random for whatever reason. So these are all the forces we talked about, like shared selection.
So it's not a 50-50 chance of you inheriting a gene from a parent if
Yeah.
you're like, because if they're together, they'll inherit them together.
Exactly, yeah. And then like what I always like to think about is if you have, the classic shipwreck example, right? So you have like 1,000 people on the ship, and then they get shipwrecked, and there's like 100 people left on the island. inheritance of alleles in the population is now not going to be random because it's such a small population.
So you're more likely to inherit those alleles
Yeah.
than to say if you were not shipwrecked.
Yeah. Well, it's also like non-random mating as well too, because when you not when you choose to mate with someone for different reasons, you're now throwing off the theory that you're just randomly inheriting alleles, right? It's now going to be different sets of alleles for different reasons.
Parents may be more likely to like be related to each other, and so you have like a 75% instead of when you have 50 C and G, like 75% chance of C because both your parents got C from the same
Yes.
common ancestor.
So think about like Ashkenaz and Jewish, right? They're marrying within a community for religious and ethnic reasons. That is going to throw off the theoretical LD because it's now not non-random inheritance.
Thank you for the clarification.
Yeah.
Thank you guys. It's really helpful.
Okay. How are we doing on Zoom? Good. Yay. I got 2 thumbs up. Excellent. Okay, so now we are gonna take the files that you either prepared yesterday or you got off of Google Drive, and we're going to estimate LD with them. So just like yesterday, make sure that everything is in the same folder, right? So you either have Plink installed on your computer or you move it to the folder. So like, I'm just gonna move mine to our day 2 folder. And then we're gonna open terminal and I'm gonna change my working directory to that folder. So I do CD space, and I usually just drag the folder name so that it populates the pathway for me. Okay, so now I'm in the folder, and this is not gonna do anything to the file. This is just going to do similar to what we did yesterday. It's gonna look at LD in our population, just so we can kind of get like a mental picture before pruning. So I will put this in the chat. And also, if you guys don't want to type from the PowerPoint, it's all in the day 2 scripts on the Google Drive. So just copy paste. Okay, so that finished on mine, and it's going to output a file called .ld. And then I drag that to my text editor, right? And it's going to result in this file. And we have biallelic samples, right? So we have the chromosome, the base pair, the SNP location, and then for the second allele, the same information. And then in this column right here, this is going to be the LD calculated for variance. And for this command, there's 2 ways to run LD. There's R squared and then just R, and R is the kind of like raw output of LD, and then R squared is the squared correlations. And I didn't really see a big difference in them when I ran both. And from what I gathered, most publications looked for R squared. So if you want to get a better idea of LD, I would recommend running the R squared. But if you look at our file, what do you notice about this column, about the numbers. It's kind of small. Do they look big, small?
Kind of big, for the most part, yeah.
Kind of big. That's what I thought too. Right? So you have some in perfect LD. So 1, which means they're always going to be inherited together. Yep, exactly, Julianne. 1. So if they're that high, they violate the assumption of LD, right? And then we're going to prune them out. And now we're going to look at our allele frequencies again. We did this yesterday. And I'll put it in the chat. And then that is going to output a .maf file. I'm sorry, .frq file right here. So I can look at that in my text editor. And as you see, I have this very large insertion, which is throwing off my beautiful columns, but that's okay. So when you look at this file, it's going to tell you the chromosome, it's going to tell you the SNP position, the first allele, the second allele, the minor allele frequency, and then the number of individuals with, or the number of chromosomes observed. So divide by 2 and you get the number of individuals, right? And what do you guys notice about the minor allele frequency column? It's smaller, yeah. But it's, the threshold I usually use is 1%, which is 0.001. So they're still a bit higher than that, but some are still 0, which is great. And so in case you're interested, the minor allele frequency is calculated by how many A1s you have out of the total proportion for this particular marker. So they basically do like an expected observed calculation. Okay, so the order that we're doing all of this QC in is kind of personal preference. Some people prune first, and then they do Hardy-Weinberg, they do kinship, they do heterozygosity, that sort of thing. I like to do minor allele frequency and LD last, because I figure why do it on individuals I'm gonna take out later? Like, it kind of defeats the purpose. But again, it's personal preference. You can also put all these commands in one long line and do all your QC just in one command. So what we're going to do today is we're going to prune for minor allele frequency of 1% or less. And then in this first command, we're going to tell Plink what to prune LD for, right? So this first number is the window size. It's going to look at 50 base pairs at a time. And then this is how much it's going to shift the window by. And then this is the R2 value we're going to prune by.
Sorry, I'm getting an error saying no such file directory B file, which when I'm running these commands.
Oh, so that's because, did you output a PED file yesterday?
From the 1.6 gig.
No, from the, when you cut the outline individuals for heterozygosity, what was the output file? Was it a .10 or a .? So use this to cut it, and then what was the output? That's okay. But you almost got there. So just maybe just get the clean files from Google Drive and go from there.
I do have the clean ones.
Oh, perfect. Yeah, so just move those in the same folder. So I think you forgot the dot dash for the clean.
I did. Comments.
Oh, you did?
That's all right.
So it looks like you didn't filter it right here. Chromosome 7 underscore filter. Yeah, so those need to be in the same folder. Yeah, and then that should work. Try it with the dot dash. It's clicking that folder. Because usually I get that flag when I can't click around. Maybe just put it in there. Okay, so I'm gonna put these in the chat for you guys. So you run that command first, Plink calculates it, and then this second command is where it actually prunes your data. So let's take a minute or two to do that. Let me know in the chat, or you can talk if that goes well, or you need any help. So I'll be right back. That works? Okay. We can look at later comments on it. If you want to enter it here. How are you guys doing?
I'm not the same time ago we did it, like we can go down.
Just to call the folder? Did you do CD? Yeah, so do CD space and then drag your folder. Is that the same? Do you have a day two folder?
Yeah, I switched to it.
So I think it doesn't like where your folder is, and I think it's because your folder is in your Mac. Is there some way to put your folder like in your Windows shell? Yeah. I think that's why it's having trouble. How's good? How are you doing?
I'm not sure what. I guess it's.
Do you have?
Yes.
Okay, excellent. And then, or maybe just copy paste it. So you're not going to be able to edit command line. So that's why I use the text editor.
I see.
Yeah. You can't like click on. And you have the chromosome in the cassette. Backspace. You have, okay, you're in the right tab. sometimes, the problem we were running into yesterday is it doesn't like spaces and folder names. So I would try and rename these single names and make it single folder again. Okay. Oh, you know what it is? You're using the Windows, so it's not dot dash. So just try the whole thing. Just start at copy and paste right now.
Okay.
Yeah. Sorry, I forgot. I'm familiar with Windows. I'm not sure if you can copy and paste. Oh, you can.
Oh. Oh.
One. What version?
It was 2.
Okay, so I think you need 1.9
Oh,
for that particular command.
Is there?
Are you guys using 1.9 in it virtually? Sure. Yeah, okay. I think that makes sense.
Okay.
So, yeah, or you can just Google download like this one first. And then, so the different version is upcoming, sometimes do different things. So try that and then see how it goes. We're gonna have another break in a bit,
Yeah.
so we'll troubleshoot then.
Yeah.
Okay, how's everyone on Zoom? Were you able to do that okay? Great. So you should be getting a log file that looks something like this, right? It's the .prune.log, and you can see exactly how many variants were loaded in. It was that little over 4 million, and we had 384 people. And then after we pruned for minor allele frequency and LD, we were left with a little over 3 million variants and same amount of people, 384, right? Oh wait, I'm sorry. We had 3 million variants removed due to minor allele frequency thresholds, which left about 800,000. And then a further 700,000 were pruned for LD, which left us about, I think, 100,000, if I'm remembering correctly.
Kelsey,
Mm-hmm.
so if we were to use the same threshold value for different data sets, then we would have to generate the LD and frequency file, right? We can just go straight into the pruning part, right?
Do you mean like pruning while you're doing the other QC steps at the beginning?
Yeah, I mean the 2 previous command that we did, right before we did pruning, like
Oh.
p-link.
Right, the checking the minor allele frequency.
Yeah, and then the LD.
Mhm.
Yeah. We don't need to generate that if we were to use the same threshold value, right?
Yeah, so these 2 steps that we did don't actually do anything to the data. It just kind of kind of lets you look at it. And if you want to adjust the threshold, it's just a good check to see what you need to adjust by. Like, say you don't want it as stringent because it would prune a lot of your markers. So you could adjust this to, instead of 1%, you could adjust it to 10%. But if you already know the threshold you want to do and it's kind of a set pipeline for your method, then you don't have to do those steps at all. You can jump right into pruning.
Does it change over data usually, or do you use like a standard threshold?
It changes, and it also depends on what you're going to do with the data. If you publish, people tend to look for a pretty like stringent threshold, and if you don't have one, then you should be prepared to justify it. But if your population size is small, or you don't do whole genome sequencing, you do like targeted gene sequencing, then you may have a much lower pruning threshold, right? And then that would make sense because you don't want to remove the majority of your markers. So like, yeah, so it really is very dependent on your data.
Okay.
But
Thank you.
this what we're doing here is I should back up. So like, obviously I use my data for population genetics. And so I'm looking at selection scan statistics after I run these QC steps. And those statistics will also put their recommended pruning thresholds. So these pruning thresholds are kind of the standard for the later statistics I would use. So I guess another thing to look at would be if you're going to run a certain test, you can maybe see what pruning they recommend as well.
Okay. Thanks.
Yeah. Okay, how are we doing? Better? I don't think I caught your name.
Oh, my name is Viet, by the way.
Yeah.
Yeah, nice to meet you. It's still fixing the error, and the version I downloaded was 1.9. But I just maybe is it like how I structure this?
It's just to be safe. Anybody wants to see all of them, but it's hard to tell, like, just to be safe. I think for some reason, just one version doesn't have that command. Oh wait. So I think it might have to be something troubleshooting with it, right? For now, I'll upload the next step files on the Google Drive, okay? So you can follow along.
Okay, so okay, so I'm gonna put them on the Google Drive for you, and they're just called pruned, okay? Okay, so now we are gonna take that prune file and we're gonna use Plink to calculate a PCA. Did you get them okay yet?
I did. I misspelled pear.
Oh, is that what it was?
Yeah, but now
Oh.
there's a new issue.
Is it in?
Yes.
Oh, so you don't have the bed file in there?
Oh.
Yeah.
Huh. Is there?
So maybe just download it again from the.
Oh wow, that's a long one.
Slow down. Oh, you don't have to download everything, that's just this small. Is this from the bottom? Yeah, the bottom. That one, that's the only one you need.
Yeah. So when you are working with the binary format, you always need the bed file, the BIM file, and the FAM file.
I see.
Yeah, and it won't run without all three. Yeah.
So what does the bed file do?
It's just like they all store different information. So like the FAM file is like the individuals, the order they're in, and the bed and the BIM are like the markers and the genotype data you're looking at. How are you guys doing? PCA, good? Okay. So, okay, so Asha, I think, are you running a Windows or a Mac? Windows. Okay, so I think, yeah, I think this is just a problem with the amount of like computing capabilities your computer has. It usually has a default amount, and sometimes you have to increase it to run certain functions. And is Meryl here? Meryl, do you have the link maybe that I sent you yesterday? I think it might be the same issue. No, I try and find it. Yes, thank you so much. So I think it's going to be a similar issue to this, Sasha, where you need to go in your settings and adjust the threads that you can request. If you don't think you'll be running this type of analysis a lot, you can just visually follow along with what we're doing. And you don't necessarily, this is the last Plink command we're going to run. So you don't necessarily need to run it if you don't need to in the future. Okay, that's another great fix. Thank you, Meryl, for suggesting. Okay, so we calculated our PCA with Plink, right? So once you get to this step, there's a lot of different methods to then visualize your PCA. And it again boils down to personal preference. So I'm gonna walk you through what I just find kind of like the quickest and easiest way for me. But if you're familiar with R, you can definitely play around and find different packages. How are you doing? Do you have a question? Good, okay. So what I do is I look for this file that it just, oh, sorry. The first thing I do is I calculate the amount of variance explained by each PCA, right? So sometimes you'll see on figures, it'll say like PC1 explains 40% of the variance and PC2 explains 12%. So the first thing I do is I calculate how much of my variance is explained. And the way I do this is I go to this .eigenval file that Plink just calculated for me. And I like to open it in Excel, so I just add a TXT extension. And then I open it in Excel, and it's gonna look like this, right? It's gonna look like a single column. And this is gonna be PC 1, 2, 3, 4, 5. So what you have to do is you have to sum all of them. And then you just divide. Let me double check. Yeah. So I do A1 divided by C, and you in Excel you have to do these $signs, C1. And you can convert this to a percentage, but basically this tells you that my first PC explains about 33% of my variance, and my second PC explains about 13.8% of my variance. Okay, so now we can take a look at the .eigenvec file, and I do the same thing. I like to open it in Excel, and then I plot it in R from preparing it in Excel. So
Could you go back to the first Excel sheet?
Oh, the eigenval?
Yeah, which formulas did you use for column itself?
Yeah, so what I did was I summed up all the columns. So this is the total sum. So just equals sum, and then I highlight all the rows.
Okay.
Yeah, and then here I do this formula. I do A1 divided by $C $2. So, or 1, wherever you put the sum, right?
Oh, okay.
And Excel is just funny where it needs the $sign between the letter and the number.
Okay. Thank you.
Yeah, you got it?
I think I'm getting there.
Okay.
How did you get the eigenvectors opened? Excel, when I open it, like they're all in one cell. How do you like?
Yeah, so and this is again personal preference, so I'll show you how I do it. I think there's like a few other ways you can do it. Like I think you could do open with, but basically what I do is I open a blank Excel, and then I go to data. I'm sure Dan has a bunch of different ways too. And then I do from text file right here, so data from text, and then I open it, and basically this forces it to ask me how I want it formatted. Yeah. And I think you can do open with and then Excel. I have a really old version of Excel, so sometimes that doesn't work. Yeah, but sometimes on other computers, if you drag it or do open with, it'll also bring up this prompt. And then I do delimited next. And then here I can see they're separated by like a space. So I do space, and then I do finish.
And then are you just saving the TXT just directly as do you call it something different?
So I will show you in a little bit, but when I plot in R, I usually save my files as a CSV so that it's easier to open in Excel later if I need to adjust something. But you can save it as a TXT and also load it in R. It's again just like personal preference, basically. Yeah. Okay, so if you go to the .eigenvec file, I like to open it in Excel. Here we have our individuals, right? We have the family or the population ID, and then we have the individual ID, and then the rest of the columns are the PCs, right? So this is PC1, this is PC2, this is PC3. If you leave Plink on default, it does 20 PCs, but you can, I think you can tell it how many you want if you wanted to limit it for some reason, like if you only wanted 10 PCs calculated. So then what I like to do is I add headers. So we only need one, right? So I call this the sample. And then we're gonna plot PC1 and PC2. So I add that. Everyone with me? Okay, so once you add that, what I do is I populate the population information. So if you remember, we pulled people from Mexicans, we pulled Yorubans, we pulled Han Chinese, and we pulled, what am I forgetting? Huh?
Wire Islands.
Yorubans, did I say? Han Chinese, Yorubans, Mexicans, and Finnish. That's what it was. So, you know, you could sit here and manually look up every single one. No, we're not doing that. So what we're gonna do is we're gonna pull up that population list we downloaded from 1,000 genomes yesterday. It's called IGSR_samples. And I open that in Excel, I just drag it. Okay, and we're gonna do this nice function in Excel that will automatically populate it for us. Okay, so I do equals v lookup, and then I highlight the entire column of the individuals I want to look up. Oops, yeah, no, I don't want to do that. So I'll just drag it. Okay, and then I do comma, and then in my reference file, I drag it over what I want it to examine. And then I do comma. And then I tell it what column I want it to auto-populate with. So in this case, I want the population code. So I'm gonna tell it 1, 2, 3, 4. 4, and then false. Because I want it to be an exact match to these sample IDs. And then I hit enter. How are we doing on Zoom? Do you want me to show you again, or you got it?
Can you show it again?
Yeah, okay. So before I do it, I made a new column, right? Called it population. I pulled up my reference file that has all the population names as well as the IDs, right? So it's the file we downloaded from 1,000 genomes yesterday. And I do equals v lookup. And then I highlight all the codes I want it to look up. So here are the sample IDs. And then I do comma, and then I highlight my reference table. So I just drag it over the columns I want it to examine. And I do comma. And then from this reference table, I want it to populate with these codes right here. So I'm going to tell it column 4 because it's 1, 2, 3, 4. So I write 4. And then comma, and then I want it to be an exact match, so I click false. And then I do enter. Guys, good?
I just want to assimilate what you're explaining, and then I'll practice myself.
Oh, okay, just absorb. Yeah, no problem. How is that on Zoom? Good? How are you doing, Asha? Were you able to? Good, Julianne. Good, Meryl. Awesome. Asha, great. Okay, so you don't need this reference file anymore. You can just exit out. And the next step is to plot it, right? Unfortunately, we don't have enough time in this workshop for me to like show you how to use R or install it. So I will show you how I plot it. And if you already have R installed, I will put my code in the chat and you can plot it yourself. Dan teaches a really great intro to R class. So yeah, highly recommend. Yeah, I think you're teaching it next quarter, Dan, right?
Yeah.
So basically, so I'm gonna save my file. Oops. Yeah, I'm gonna call it, I don't know, chromosome 7 PCA. And I usually save mine as CSVs, but again, personal preference. And then, okay, so in R, I'm gonna call up my working directory. Oops, this is not my right plot.
So you save that Excel sheet as
Um,
7 PCA CSV?
Yes, I did. Okay, so I usually plot, I'll put this in the chat for you guys. Oh, is this the, please explain again for the VLOOKUP, or is that old? Old, okay, thank you. You guys are on it, I appreciate it. So I like to plot in ggplot, but because I think it's aesthetically like a little bit cleaner, but it's up to you. So I load my data, I install the library, and then basically I'm just calling it a plot. I load ggplot, my data. I want to plot PC1 and PC2, and I'm coloring by those population codes, right? So if we do that, voila, we have our beautiful PC plot. Up here. And this is the wrong code, so ignore this, guys. And it should look something like this. Right? So what do you guys notice when we look at this PC plot?
There's a mixture between Finnish and French.
Do you mean Mexican? Yeah, sorry, the words are really small. Yeah, yeah. So, well, historically, we know that there was, right, colonization in the Americas. So we would expect some European admixture in American populations. That being said, if we had pulled maybe like Spain Spanish instead of Finnish, we would probably see a lot more overlap between these populations. But obviously Finnish are, you know, European, so you do see that clustering you talked about, right? But we see that the your Rubens are way off by themselves in grouping together. Do you guys, anything else? Oh, in the chat. Yes, Julianne, exactly. There's like these huge, right? It almost looks like the plot I was showing earlier. At least to me, that's what it looked like. It looked like this, right? These huge kind of gaps between these distantly related populations until you introduce Mexican Americans. What? Too far. Is there a reason why the PCA? I think it's favored, PCA is favored because it's a visual way of reducing a lot of different dimensional data in individuals. And I think when you're working with a lot of markers and you're working maybe with a lot of individuals in a lot of different populations, it's a very striking way to represent genetic diversity. And once you get the hang of this process, it's pretty easy to, right, to plot a PCA. So it's a pretty accessible method. That would be my best guess as to why it's so popular. But I think a lot of people in other social sciences use PCAs to look at qualitative data as well. It's not just for genetics. Yeah. Okay, so we're gonna take another 10-minute break. It's a very chill day today. And then we're gonna do our admixture plot with these same individuals. So we'll come back at, let's say, 10:45. Yes.
So, I don't have the file, so it said it's been written to CH7 pruned and pruned in and prune out. I don't have the so this is my day 2. I don't
Yeah.
yes, but the extent is that the file that you put into the Excel sheet, the dot.
Oh, so you want to do the second command. So what was the second line? Okay.
Because
So in that folder, look at the day 2 scripts text file.
oh, so that's where everyone's been getting the code. I've been typing it out.
Oh no, don't do that. No, that's way too tedious right there. Yeah, so that's all the scripts we're doing today. So you just need to do the second step.
I see. I've been like that's why it's been taking so long.
Oh yeah. So in your case, you can copy paste that whole thing, but just don't copy paste the dot dash.
So this would.
So this basically prepares the file to tell the program what to prune for, and then this second command prunes it. So you did the prep phase, now you need to do the prune phase. 6, 5, 8, 2, 3. Yep, there you go. And then the next step. Oh. So why don't you rerun that first command too? Just copy paste that.
Change the 3, 5, 8 to 3.
Maybe there was like a so I have, it's so sensitive to potatoes. So now it's happy. So now you can do this one. There you go.
I just like watching it load.
I
like,
know.
It's so satisfying.
It's very quick. So now if you look in your folder, right here.
Should work.
If you reload it.
Yeah, there you go.
And then just
So the
one being stuff
dot
about it.
eigenvalue is that single column where we calculate the variance. And then this is the one where I added all the eigenvec. So that's your eigenvector coordinates where it's like reducing the dimensions. That's the one that I add the headers and I plot the PCA.
Okay, so let me just do that real quick.
So I add txt to force it to do that tab delimited. But since you have Windows, it very nicely opens for you. So I'm very jealous. So then I just add all these.
Oh, I need to put some. Sorry, I'm more of a Google Sheets person.
There you go. And then I do equals and then A1, right? No, just one at a time. So I do A1 divided by $sign. You have to do $sign. C, you just type it in. So C, $sign, 2, or 1, yeah, and then enter. There you go. And then double click that little button, or yeah. So you can't drag it, you have to double click that little box. Oh, I forgot the $sign.
Oh.
So it's $C, $1. So just move the $sign over. There you go.
What is the $sign?
It basically tells the program that the column letter and the column number are separate data.
Oh, so
It's
it
just
doesn't
an
like
Excel thing, yeah. So just enter. And then now you can double click that little dot. And this is the percentage of variance that's explained by each row. So your first PC will explain 33% of your population variance, and your second PC will explain 13.8%.
And then after that, we
So open that in Excel too, and I'll show you on the screen. Hey everyone on Zoom, we're still on a break. I'm just gonna go over the PC again. So you add, right, txt, and then I open it in Excel. And I don't know if you have to do this on Windows, but I have to tell it it's tab delimited. And then I separate by space. So I check the box and I hit finish, right? And then we just add the headers so it can plot in R. So each column are the different PCs, right? You can go all the way if you want. And then this is the individuals, and this is the population level. In our case, they're exact same, so I don't need to label them differently. Oh. Did the program not okay? Your computer is working really hard, so I almost wonder if it's because you're trying to plot in your shell program. So I might exit out of that and do an R install just like on, or only through Windows. Oh, it is? Okay. Because you're still in your Windows though.
that's what I'm saying, sometimes when you try and do these in your shell program with Windows, it takes a lot more out of your computer. So I might like switch to plotting it on your Mac.
Oh,
makes
so I don't
sense?
have it on my mind.
Okay,
Yeah.
okay, it might be, that's why it's really slow and worrying and getting caught and stuff, yeah. Because I have parallels on my
Oh, yeah,
computer
parallels,
too, and it's, yeah, when I try and do like some of the programs,
If it's like,
how are you doing? Are you at that step?
Yes, I am,
Okay.
in a column.
So add PC, oh, so you need to open it, in a different way so that all of this data is in separated columns. So exit out and just open a blank Excel. Oh, so what you need to do is go to your folder and add .txt. You can change the file extension. So you can just, yeah, rename and then dot. Nope, you're fine, you can leave all that, that's fine.
Oh.
Yeah.
Oh.
So just literally type .txt. No, at the end. So itembank.txt. There you go. Yep, that's fine. And then, yeah, it changed it right there.
Oh, okay.
So then go to Excel and I do get data from text right there, so that button. And then open that file. Yep, that one, that one. And then
For a
import.
second, I was wondering,
Okay, so now
delete the
you need
PC
to
one,
tell it
PC2 really
that's
quick.
perfect and hit
Just-
load.
Like not the whole thing, just those
Right, so now it told it to be in
Yeah,
different
delete
columns.
that temporarily. Highlight column
And then you
D,
need
so
to
just
relabel.
go to data.
No, yeah,
text
so,
to column,
and then individual, and then PC1, PC2.
next, space, finish.
Yes.
Okay.
So then, you're gonna find this table from yesterday.
The TSB.
Yes, the TSB, exactly. And
Oh, okay.
this one.
Do we drag it?
Yeah, I just drag it. I mean, you can
Text to column.
open it however you like.
Next,
And I just have it open, right?
space, yeah, finish.
And
The
then,
issue was that this was all this data was all in one column and it was
okay, so in your blank column,
and then add back the PC1, PC2 names.
Yes.
I do equals VLOOKUP, right?
Yes.
And then I drag it over all these individuals that I want it to look up.
Just give it
Just the individual, not the population.
oh, yeah.
Either one, the codes are exactly the same in this instance, so it doesn't really matter. But just make sure you drag it over the whole column.
Yes.
Yep, and then be careful.
let's try that.
Okay, so you don't want to start at the header, you want to start at the first data value. Oh, you did, perfect. Okay, so comma.
I think yours is just different. So then, and then I drag it in my reference file, I drag it over the columns I want it to reference. And I do comma.
So the first one.
Yeah, I mean,
Oh, wait.
I just drag it over all of them, but we want the fourth column, so technically you could only highlight that column.
Comma.
Yeah, so then comma, and then it's column 4. So 1, 2, 3, 4, so 4, and then comma, and then false. And then enter. The reason I drag it over all of them is sometimes I want to populate it with different information, and then I don't have to do VLOOKUP again. I can just tell it like column 2, right, if I wanted sex, or I could tell it column like 6 if I wanted continent information instead of country information. So all you would have to do in this formula is change the column number, right? That's kind of why I drag it over all of it, but
That's really, really useful.
yeah. And then, yeah, now you have your population information and you're ready to plot in R.
Isn't spell?
Huh?
So if we look up individual ideas are.
So I wonder if it doesn't like that individuals. I'm not as familiar with. So is the header highlighted? You don't want the header highlighted.
So,
Yeah, you have to do, try it again.
yeah.
Yeah,
Okay.
so that's what you want it to look like. Okay, and then comma. And then, oh, so I just go to the header and I click and then I drag like this.
Oh,
Okay.
I don't know, good old.
and then try more. Spell.
I wonder if it's because your headers have like a drop down, maybe the format got weird.
Multiple results.
Maybe just try, it might be, is your Excel normally like look like this with the drop down?
No, not
Okay. I
really.
think it might be because it opened it weird a little bit. So maybe try and do it in like Google Sheets or somewhere where you're more familiar.
Mhm.
I think it might be because it, yeah, that's
Maybe if I
concerning me.
just type it physically,
The
the number, C2 to
C385.
Okay.
Oh, there's like, feels good. There's a few good. That.
Still spelling.
Okay, well, let's troubleshoot it after we,
Yeah, definitely.
yeah.
That's your skip.
Okay, so our next step is we are gonna download this admixture program, and it's exactly like Plink, where it runs the same way, right? It's a really quick download, and then you just put it in your working folder, or in the future, you can like install it on a Conda environment if you just wanna call it locally.
And are there any programs like that you ever use for PCA?
I think there's some way, you mean instead of doing it in R?
Or instead of doing it manually, like an art package?
Oh, I mean, I think there's a lot. Yeah, there's none that I particularly use. I've done it before in Python once, but I think there's a ton of R packages that just do PCA. Yeah. Usually what I do is I plot it in R and then I adjust all the like colors and I make the font bigger and all of that. Yeah.
Yeah, my plot shows my plot is not showing very well. It's like my axes are up completely.
That sounds like a
Do you know how to manually set the axes? You could go,
This is my Excel.
there's a stories data point. So if you can force the axes to be easier to remove.
Oh, my
Did you
God.
change all the headers in your Excel? So you are calling it something different? Wait, no.
I see that one here, you just have to find like this like there. It's very odd. Well,
I don't think it's, I need to the Excel file, I don't think well.
I guess, even if we just set the line limit to we can like
The
pretend,
axes.
we can pretend that.
Yeah,
like,
that's
doesn't
very
exist.
strange. I think we have to figure out why your R is defaulting to that. Because it should detect the axes and then adjust it.
Well, it's because there's one spreads data point, so it's forcing the axis to do this, but if you could force the axis to be 0.07, then it should plot normally.
But we should all have the same data.
Yeah, so somewhere in Excel, we could like hit a button.
Oh, maybe we deleted, like a
Yeah.
decimal point somewhere, I guess. I'm guessing like a decimal point got deleted.
Maybe, so you're just plotting PC1 and PC2, so maybe what I would do is I would sort these two columns, largest to smallest, and make sure there's not something weird like a negative that got deleted, right?
Okay,
Because PC1's all negative, so if you have like a large positive, it might be
okay.
freaking it out. Okay, for you guys, that's the program? Yeah? On Zoom, do we have the program downloaded? Yes, Emma.
I think it only works for Linux and Mac. They don't have Windows version. Right?
Oh, they don't.
Oh, really?
I think you might have to use structure. This is another program that a lot of people use. Unfortunately, I'm not as familiar with it, but the concept is exactly the same. So you'd probably just have to play around a little bit with how the data needs to be formatted to plot with it, but it should take the same data we're about to do. I didn't know that. So for those of you who have Windows and it's maybe not working, just follow along. And we're gonna do the admixture analysis in Plink, and then how you plot it later, you can try it with structure in the chat. I think there's also a couple other programs with the Broad Institute at Harvard. So this is just one example. You don't have to do it like this. So the reason that I personally use admixture is because it has this really interesting function where you can calculate the cross-validation error in order to find the ideal k-value for your populations. So if you think back to this plot, right, the researchers told this program, I want 5, sorry, this is in your guys' way. I want 5 ancestral segments for these individuals, right? So it's not exactly unbiased. You are telling the program to make 5 ancestral segments. And in my own data, this actually had a pretty large effect. So here I was looking at Peruvians, and I told the program, okay, we have this a priori knowledge. We know Peruvians have European ancestry. We know they have East Asian ancestry from, early indigenous populations crossing Beringia. We know that they have indigenous American ancestry, obviously. So That's 4 different, oh, and African ancestry. So that's 4 different ancestral segments. We used our prior knowledge, and then I looked at my individuals to see their ancestral proportions. But what I also did is I also calculated the ancestral proportions that the program thought would be most appropriate for the data that I had. So the program actually told me that 9 ancestral segments would be most appropriate. And I did it using all the populations on 1,000 genomes, and it actually subdivides them for you. So it basically told me 9 is the most appropriate, and then this is how you should group it, which is interesting. So it did Southern European versus Northern European, West African versus Central African, East Asian in 2 different categories, South Asian, and then it had Peruvians and other Indigenous Americans. But taken overall, you can see that the ancestral proportions do shift depending on how many components you tell the program to run. And it's the exact same way in structure, right? The other program I mentioned. So in particular for my individuals, you can see that the European ancestry for these 2 almost completely disappeared, or actually it did completely disappear, right? And for this individual, here it was picking up East Asian ancestry, and now it's disappeared. So what this tells me is that most likely the East Asian ancestry that has all but disappeared in this different reconfiguration, this is probably just an artifact of the statistical program and not a true ancestral representation. So again, my larger point is the ancestral proportions can greatly change depending on the settings you're putting in your algorithm. And in order to calculate the ideal amount of ancestral proportions, I've run this code in admixture, and I basically tell it, look at 1 through 10 different ancestral proportions in my data set, and then output the cross-validation error. And I want to look for the ancestral proportion with the lowest error. When I ran this, it took 3 hours. So we're not going to do this in class, but this is why I personally prefer admixture over structure, because I don't think structure has this built into the program. I think you have to basically build your admixture plot on prior knowledge alone. I don't know if they've done an update with structure. I'll have to investigate that further. But when I ran it, it spit out a log file for each K, right? So that .log value. And then each K has what's called the CV error, the cross-validation error, and I threw them all in an Excel here, and then I plotted it, right? So when we look at the cross-validation error for our data set, it's very clear, I think, which K value is the best, right? It's like, this is like one of the most dramatic ones I've seen, where it's like, oh, so, and what you're really looking for if you do use admixture and you do want to plot this ahead of time, which I recommend, is you want to put enough K values in the algorithm that you see it go back up, right? And that way you know for sure you've reached the lowest error possible. Because if you're getting lower and it just keeps going, you don't know that you've reached it. So I usually start with 1 through 10, and that's usually sufficient.
So in this example, the K value of the best CV error is going to be 3.
3. Yeah. So 4 is also okay right here, but you can see it slightly goes up. And I mean, you don't have to plot this. You could just sort it in Excel and look for the lowest CV value. I like to plot and visually look at things, and this to me is a much clearer, okay, 3. But you can see here that 3 and 4 have very marginal differences in CV error. So this script is in the day 2 scripts on the Google Drive, but again, it's gonna take a few hours and your computer's gonna get hot and whir a lot. So I recommend running it overnight. I recommend running it not on your personal computer if you like to watch TV on your personal computer. Yeah, so we're not gonna do it in class, but we are going to run admixture. So this is the program, right? I'm sorry, we're not running it in Plink, we're running it in admixture. That's my bad. So if you don't have admixture, I will try and figure out what the structure command is for you guys. But basically you call up the program, you load in the Plink file, I tell it how many K values to do, and then how many computer threads you want it to run with. And that just makes it run faster. So you can just put in like 1 or 2 if your computer is maybe older or doesn't have a lot of threads. So what you want to do is make sure in your working directory you've dragged that new admixture program. So I have my pruned, I need my bed, my BIM, and my FAM files all in one folder. Here they are. And I have my admixture file. And I think it'll take about a minute or 2. So while that runs, oh, and then another input that I usually do is I usually flag, I think it's at the end, or it could be at the beginning. This flag B, this will add about 250 bootstraps so that the eventual admixture plot has a higher confidence level. Again, this takes about an hour or 2, so we're not going to do that in class, but I put this up here because when you run it on your own, you probably want to do bootstrapping. So while that runs, check. So Asha, if I were to run this on Hoffman, I don't know if Hoffman has this installed. You might want to like install it on Conda on Hoffman, and then I open an interactive session, and I would probably do it from there. Yeah, most of the stuff I do on Hoffman isn't pre-installed, so I have to Conda call it. Let me see. Yeah, so I would call up Hoffman, and then Conda install it, and it should run just fine. You don't need Conda to install. Oh, on Hoffman too. I think that they could install it on Hoffman, but you'd have to request it. I think you'd have to put a ticket in, and it's just faster to call it in a Conda environment. But Conda is preloaded on Hoffman, so you don't need to install Conda, which is why it's easier. I honestly, I don't even run admixture on Conda because as you can see, when I don't bootstrap, it's already done. If I bootstrap it, I ran this on like 3,000 individuals and it took like 2 or 3 hours. So it's not that computationally expensive, I think. I usually save Hoffman for like overnight jobs. Bootstrap. That's a good question. And I don't think I'm going to do it justice, so I'm going to Google it for you guys. Yeah. So basically, you're running it over and over and over again in order to have higher confidence in the eventual output, is I think the really very simplified example. And then there's usually like a bootstrap threshold where the error rate reaches a very low amount. Yeah. Good question.
It says admixture's not, not that they're downloaded, but in the same. Oh, right.
Yeah, sorry. I'll add that to the PowerPoint. Did it work for you? Okay, Tina? Yes, great. Okay. How's everyone on Zoom doing? Because now I'm going to show you how I would plot it. And we should finish early today, so I can stick around for a little bit and look at structure if people are interested and try and figure out how you would install that on Windows and maybe play around with it. So if you're ready for next steps, give me a thumbs up and I'll just show you how I would plot the output in admixture. Thumbs up, great. Good. Okay, so it's really similar to how I plot the PCA. Basically, you look for this output file. It's going to be .q, and the 3 is the ancestral components you told it to do. So I go ahead and I open that in Excel, just so I can get this delimited. Columns to pop up. And these are the 3 ancestral components and the percentages that belong to each ancestry. So for our question, we found from our PCA that the Mexican individuals had a bit of shared ancestry, right, with the Finnish European, and maybe we're clustering a bit closely to the East Asian. So for our admixture plot, we are going to find out for the Mexican individuals what proportion Finnish, Mexican, and Han Chinese and African ancestry that they have. So in order to do that, this is where the order becomes important. We're gonna go back to this pruned binary file with the .fam, and this is gonna tell us how the admixture program took the individuals, like in what order. So I go to my pruned fam file, and I open it in Excel. Okay, so now we have our population and our individual identifiers, and I'm gonna add them here. So I just copy paste it to preserve the order. So it should look like this. Oh, I guess I changed it over. There's no preference. I guess, so for 1,000 genomes, these are the same numbers, right? They're duplicates, but sometimes they can be different. So it just kind of depends. And then we're gonna do the same thing. We are gonna do the VLOOKUP to pull the population information so I know what I'm looking at essentially. So I'm going to pull up my IGSR samples TSV and I just have it in the back. And then I do that exact same process. I do equals VLOOKUP, and I want to look at either column, it doesn't matter. I do comma, and then these individuals, comma, column 4, and false, and then enter. And I'm just going to quickly add some like headers, right? Okay, I'm going to pause there. How are we doing? Okay,
Oh, I'm just waiting for my.
thing to populate.
Yeah.
How are we doing on Zoom? Any questions, concerns?
What was it, Carolyn?
IGSR. Yeah.
With the
Oh, so this one is going to be the file that we just made that has the .q. Yeah. Asha, do you mean this one or the PCA one or the one we just downloaded from 1,000 Genomes? This one. There in the background. Yeah, so I do, I'll do it here too. I do equals VLOOKUP and then all these individuals. And then I just kind of highlight the whole thing. And then for this, I'm pulling the population code, column 4. Depending on what you want to do, you could pull the continent code, right, which would be column 6, or you could pull the full name, column 5. And then I do false because we want an exact match. Sorry. Yeah, so the reference file we pulled yesterday, it's from 1,000 Genomes. So I can show you where to get that. So when you go to this website, it's going to take you to this page and you're going to click filter by data collection and then 1,000 Genomes phase 3 release and then download the list. And all of these steps are in the PowerPoint from yesterday, which is on the Google Drive. So if you want to revisit that or see the steps more slowly, I would recommend checking that out. Yeah, great. Okay, so hopefully you're all at this step. we have our population codes, we have our individuals. So now this is kind of where it gets a little, you have to use your human best judgment, right? So what I do is I look at the distribution of the percentages. So right here, it's a very high percentage, right? It's like 99%. And if you scroll down, it stops right when it transitions from Finnish to Yoruba. So I'm going to call this column Finn because this represents Finnish ancestry. And then when I look at this column, it's clumping heavily with Yoruba, right? 0.99998. So I'm going to call this YRI. And then in this last column, it's clumping heavily with Han Chinese. So I'm going to call it CHB. And this is, like I said, where you kind of have to use your human best judgment, because if you're running like 10 ancestral proportions, say for instance, in my data, right, it's a lot of different populations that are clumping within these codes. So you kind of have to call them appropriate names. So I came up with like Northern European, because it was like Finnish with, I don't know, Norwegian or something, and versus Southern, which is more the Iberian Peninsula. Okay, so because I plot this in R, you have to slightly reorganize the data, and it's not that complicated, it's just kind of tedious. So what I do is it has to look like this, right? It has to be, at least the way I load it, it makes it easier. So one column of the percentages, one column of the population, and then the individual IDs are going to be repeated 4 times. So because we're interested in the Mexican individuals, I'm going to scroll down to MXL right here. And then here, I'll freeze the, okay, so I'm going to take the individual IDs and Yoruba, and I'm going to copy and paste this here into a new sheet, and then Yoruba. Oops, Yoruba. Yeah. Okay. And then I copy all the Mexican individual IDs again. I put them in the same column. And now I'm going to take the Finnish proportion of ancestry. I just copy and paste, same column, and Finn. And then the last one is I'm going to take Han Chinese. Copy and paste it. Okay, so you should have 3 columns. The Mexican individual IDs should repeat 3 times, and then you have the different proportions of ancestries next to them. And if you're more comfortable with loading the data in a different way, again, it's all personal preference. So I just call it population, percent, and individual. So I add new headers. And then I'm gonna save it as chromosome 7 admixture. Okay, now it is ready to plot. So this is the code I use to plot in R. And I covered 7 admixture. And then to run it, I just highlight the whole code, right? And run. And I added a few code in here to rotate, because our individual IDs are kind of long strings of numbers. This code just rotates those IDs, right? Because otherwise they would be horizontal and they'd all smear into each other. So I just rotated it here. And I added a title. We're looking at Mexican individual ancestry. And if we look at our plot, maybe it's easier on here, we can see the x-axis is each individual, and then the y-axis is the percentage of ancestry, and we have our legend to the right of what each color or what ancestry each color represents. So what percentage of, say, like Finnish ancestry they have, Yoruban or Han Chinese. So for those of you that are able to plot it, or those of you who are just watching along, when you look at this admixture plot, what are your thoughts? Because when I made this, I had a very strong first thought when I looked at it.
Like, it's just overwhelmingly green.
Yeah, it's overwhelmingly European, but they're Mexican. Why is that weird?
This is a concept of founders population. Not quite, yeah.
Almost, yeah. You're almost there, like you're very close. Yeah. Well, think about it this way. They are American populations. So what would you expect their highest ancestral proportion to be? It's not a trick question, I promise. Anyone in chat?
Native American.
Yeah, I would expect them to have higher indigenous American ancestry, right? So why don't they?
Well, this is like a pollution, like it is, the options were like Chinese, Finnish.
It's exactly it. The reason they don't have it is because we told the program to only look at 3 ancestries. So it cut indigenous American in this instance. So this is an instance where I would go back and rerun my program using 4, because it had similar cross-validation error, and it would make more sense for these individuals, right? But if you tell the program's not wrong, right? If you tell the program 3, I would also expect these individuals to have more European ancestry in comparison to African ancestry, right? Because of what we know about admixture events in these populations. So the y-axis is scaled to 100, but it's just in decimals, 1.00.
So what's your name with columns again?
The columns. I called them individual, percent, and population. You can call them whatever you want. You just have to tweak the R code. Yeah. So what you can do, if you don't want decimals, is you just look up the R code to relabel the y-axis and make it say like 100%, 75%, that sort of thing. But yeah, so I guess this is just my point of like, it's great that the machine calculates these things for you, but you also kind of have to use your best judgment when you use a lot of these programs. Yeah, Julianne, that's a good question. So if you wanted a fourth category of other, you would have to go back to the admixture program and tell it to do 4 ancestral clusters instead of 3. So because we only told it 3, it's only gonna give you data for 3. So you wouldn't even have an other at this point. Yeah. Mm-hmm. Yeah.
But if you wanted to have an other for the subcluster, could that subcluster include like a variety of different regions instead of just like one place?
Yeah, it could. It just depends on the populations you put into it, right? So if you wanted to look at these individuals on a greater level, I would probably import more individuals from 1,000 genomes, and then it would most likely, instead of just Han Chinese, it would most likely cluster like Han Chinese with other East Asian populations, right? So then you would have more of like a East Asian ancestry instead of like a country Han Chinese ancestry. So we're just doing a very scaled down proportion of it. Yeah. Yeah, but so you can do that. You just have to put more data into the program. That's the short answer. Yeah. Any other questions? It's really hard to bring up the. Okay. So that's it for today. The take-home exercise, if you do have R, or if you don't have R, again, I recommend downloading it. If you do have R, try and go back to that Excel with the PCA and plot PC1 versus PC3 and PC1 versus PC4 and take a peek at like, how do the populations shift? How do they shift? Do you think like one plot is more appropriate than the other or not? I'm gonna upload this for you guys, so no stress. If you don't have R or you couldn't get it to work, don't worry about it. We're gonna review this exercise tomorrow in the morning. So we'll look at the plots and discuss then, but it never hurts to get more hands-on practice. Yeah.
This can be cool, or I feel like when I went
Yeah,
over
one
was my
second.
Excel, my plots or my.
Yeah.
Yeah,
And then
that's-
let me just finish up with Zoom here. And then guys, one other program I would recommend, it's called HaploView. And oops, sorry, that's my text. And it basically plots the linkage disequilibrium in your individuals. So we pulled it up in a table and we looked at it that way. But if you want a more visual representation, a lot of people will use this program to visually represent it and then put it on like a poster in a paper or that sort of thing. So yeah, that's it for today. I'm gonna help the people here in person with a couple troubleshooting, but if you were curious in structure, stick around and I'll try and troubleshoot that as well.